So here we'll look at DNA replication, including all of the steps and the key molecular players that help with the process. So DNA replication is a semi-conservative process, and that means that during replication, the two strands of the helix separate, and each one acts as a template for the synthesis of a new complementary strand. And it's the parent strand that instructs the synthesis of that new daughter strand using those Watson-Crick base pairing rules. And the reason that this is called semi-conservative is because after replication, each of these new strands has one old or conserved strand and one new complementary strand. So now let's look more closely at the process of DNA replication, and we'll start with the origin of replication. So in this image, we're comparing the origin of replication in a prokaryotic chromosome on the left and in a eukaryotic chromosome on the right. So in prokaryotes, replication initiates in one place. So there's a single origin of replication, and it's defined by a specific DNA sequence that's generally rich in adenine and thymine. So first, several sequence-specific factors bind at that origin to initiate the local unwinding of the DNA, creating this replication bubble and two opposed replication forks. So replication proceeds in this bidirectional manner from the origin, so there's replication in both directions, and it continues until the forks meet. In eukaryotes, DNA replication is a little bit different, and this is necessary because of the large size of eukaryotic genomes. So eukaryotes solve this problem by having multiple origins of replication. And so this allows the opening formation of many replication forks, and then replication can continue until neighboring replication forks meet and the entire genome has been copied. So for each division, cells have to duplicate their entire genome exactly once. So each one of the thousands of origins in the genome is instructed to initiate replication only when the cell is ready to divide, and every one of them initiates replication only once. Now let's take a closer look at what's happening in these replication forks. So here's a close-up view of one of those replication forks. And we've said that to start replication, the DNA duplex has to be separated into two single-stranded templates. And so the replication fork is just the point of transition between this double-stranded molecule and these single strands. And there are a few things that are going on here. So first, the Watson-Crick base pairs that form this double helix have to be broken apart. And the enzyme helicase does this. So helicase is what translocates along the DNA duplex, unzipping or unwinding the parent strand and leaving these two single-stranded templates behind it. So there are many helicases in vertebrates, and they're involved in DNA replication along with transcription and DNA repair. So once unwound, we're left with these two single-stranded DNA molecules. But single-stranded DNA is generally very unstable and will try to form base pairs in whatever way it can, maybe by re-annealing with the strand it was just separated from, or by binding to itself. So to stabilize that single-stranded DNA and prevent it from re-annealing, single-stranded binding proteins bind to that unwound DNA. And finally, progressive unwinding of the helix in one region, like at the replication fork, causes overwinding everywhere else. So in theory, this could be fixed by end rotation, but because of the way the chromosome is organized, DNA ends can't freely rotate. So when one region of DNA is unwound, there's overwinding in these regions where the strands aren't yet separated. And then as the replication proceeds, there will be more overwinding, and the helix would start to form very tight coils and knots, and this is called supercoiling. And then as the degree of this supercoiling increases, it becomes harder and harder to continue to unwind the helix, and then DNA replication is blocked. And so topoisomerases are enzymes that control the degree of supercoiling in genomes by introducing these transient strand breaks. So they bind to the backbone, break a phosphodiester bond in the DNA strand, allow the DNA to relax, and then as topoisomerase leaves, the phosphodiester bond is reformed. And so there are several types of topoisomerases. Topoisomerase 1 introduces this single strand break, and that allows two sections of the helix on either side of the break to rotate freely relative to each other, using the opposite strand as a swivel point. And that allows the relief of tension that has accumulated in the wound DNA. Type 2 topoisomerases introduce a double-stranded break in the DNA, and so this break is like opening a gate. It allows another nearby double helix to pass through the opening, and then it reseals the break and dissociates from the DNA. Topoisomerase also has clear clinical relevance, so cells need to unwind DNA to grow and divide, and by targeting this process, you can target actively dividing cells like aggressive cancer cells. And so some of these drugs specifically inhibit this repair step. So topoisomerase would introduce a strand break but wouldn't repair it. So it would leave behind this lesion that's really difficult to repair with another cellular mechanism and is therefore lethal. So topoisomerase inhibitors can be effective chemotherapeutics. So now let's look more closely at the process of actually creating that complementary DNA strand. And so DNA polymerase is really the engine of replication machinery, and it catalyzes the addition of new nucleotides to those new complementary strands of DNA. But there are a couple of characteristics of polymerase that affect how replication proceeds. So first, DNA polymerase can only add nucleotides to an existing piece of nucleic acid. It can't start a new strand of DNA from scratch. And second, DNA polymerase can only add new nucleotides to the 3' end of a nucleic acid strand. So because DNA polymerase can only add nucleotides to an existing strand of nucleotides, a short primer is laid down here on the template strand, and the primers are shown in yellow. And so primase is the enzyme that uses ribose triphosphates, or NTPs, to lay down these short RNA primers. Once the primer's laid down, that's when the DNA polymerase, shown here in red, can begin adding additional complementary nucleotides to the 3' end of the primer strand and move down the strand of DNA. And so here we're looking at an example of a prokaryotic replication fork. So polymerase 3 in red is the primary enzyme involved in replication, while polymerase 1 in purple is involved in excising those RNA primers and replacing them with DNA. So we just saw that DNA polymerase functions to add new nucleotides onto the 3' end of a growing DNA template, and it matches the bases using those Watson-Crick base pairing rules. And so here we're just looking at a very close-up view of that happening. And this green DNA polymerase is adding complementary bases. And as it does, it's advancing, moving up the strand. And in general, DNA polymerase does a pretty good job of matching the correct bases. But sometimes it screws up. And so what happens if that DNA polymerase makes a mistake and adds or pairs the wrong base? So that's shown over here. Some DNA polymerases, including prokaryotic polymerase 1 and 3 that we just saw, also have an editing or a proofreading exonuclease activity that resides elsewhere on the polymerase protein. And so the exonuclease activity acts in the reverse direction of synthesis, or 3' to 5', so that if DNA polymerase accidentally matches an adenine to cytosine, for example, it can excise that mismatched nucleotide and give polymerase a second chance at adding the correct nucleotide. So this exonuclease function increases polymerase's overall fidelity, or how often it inserts the appropriate nucleotide. So in both prokaryotes and eukaryotes, there are many different polymerases, and the nomenclature can be kind of confusing. So in mammals, there are about 15 different polymerases, and all eukaryotes primarily use 3 of these different polymerases for replication of the genome. So now if we look at the polymerases, and Pol delta is in dark blue down here, and it has an essential function on the lagging strand of the replication fork, and it's also involved in DNA repair processes because it has that editing exonuclease or proofreading activity we just talked about. Pol epsilon in teal plays a major role in the leading strand DNA synthesis and also in nucleotide excision repair with its exonuclease activity. And you'll notice that both of these polymerases, delta and epsilon, are associated with that sliding clamp pCNA molecule. And it's like a donut that circles around the DNA behind the polymerase. And so it's just to stabilize the polymerase, binding to the template strand and increasing the processivity, or the number of bases synthesized per binding event. And so finally, Pol alpha in lighter blue. It's a little different, and it usually exists in a complex with primase, which is shown in yellow here. And it's involved in the initiation of replication, but it has limited processivity, so it has a more limited role in replication. So now let's talk about the differences between the new growing strands on the top and on the bottom. And so you'll notice that the DNA duplex is anti-parallel. And because DNA polymerase can only add nucleotides to the 3' end of a DNA strand, one strand will grow continuously in the 5' to 3' direction as the fork continues to open up. And so this is the leading strand. While on the other strand, synthesis occurs semi-discontinuously in the opposite direction in these multiple short segments that are joined together later. And so this is called the lagging strand. And so DNA synthesis along the leading strand is pretty straightforward. Primer is laid down and synthesis is initiated, and it continues along the strand. But the situation with this lagging strand is more complex. So a primer is laid down and initiates synthesis that continues until it meets up with the 5' end of a primer from a previous segment. And so that creates these DNA segments, which are several hundred nucleotides, and they're called Okazaki fragments. And so these Okazaki fragments have to be resolved or joined with the help of DNA ligase before synthesis is complete.